Well, good evening, everyone. I'm Keith Martin. I'm Director of the Centre for Eye Research Australia and Ringland Anderson Professor of Ophthalmology at the University of Melbourne, and I'm also Chair of CERA, which is CERA's not-for-profit clinical trials centre. So I'd just like to start by acknowledging the traditional owners of the land where CERA and the Eye and Ear Hospital are located. That's the Wurundjeri and the Bunurong people of the Kulin Nation. And I'd just like to pay respects to elders past and present and to their families, and also extend a welcome to our Aboriginal and Torres Strait Islander peoples who are here today, and any other First Nations peoples from around the world who are joining us in person or online. And ahead of next week's Closing the Gap, National Closing the Gap Day, I think it's important to acknowledge that First Nations people are disproportionately affected by vision loss and blindness at a much higher rate than non-Indigenous Australians. And so the latest figures show that around 11% of Indigenous Australians have vision loss in both eyes. That's a rate almost 3 times higher than non-Indigenous Australians. And that's really an unacceptably high gap. And I would like to highlight the efforts of CERA researchers and many of our clinical colleagues who are working to close that Indigenous eye health gap. And especially CERA's founder, Melbourne Laureate Professor Emeritus Hugh Taylor, who's with us here this evening, who also established the University of Melbourne's Indigenous Eye Health Unit. So just before we begin, a couple of housekeeping matters. So the toilets are located behind the auditorium, so you exit the door here, go along the corridor through the area and around to the right. And there are additional toilets by the lifts. And then in an emergency, which is extremely unlikely, again, head through the doors, and there's also some doors at the top of stairs at the back of the auditorium. And make your way down the stairwells if evacuation is required. So it's a great honour to have so many of you here tonight for the 2026 Gerard Crock Lecture in the middle of World Glaucoma Week. Glaucoma is the world's leading cause of irreversible blindness, and finding treatments for glaucoma is a major focus of tonight's lecture and of my research program and other research programs at CERA. So I'd like to welcome some of our honoured guests tonight. First of all, Victoria's Chief Scientist, Amanda Capels, who is with us, Melanie Eagle, who is the chair of the Eye and Ear Hospital and a member, and also to Barinder Giddy and members of the hospital's executive team. We also have directors of CERA here tonight, and Suwani Dharmalingam and Professor Andrew Cuthbertson, who is also Vice Chancellor of the University of Melbourne, and Simon Bruin, who is also director of the Eye and Ear Hospital. We have Colleen Lewis from our Consumer Advisory Group, and we also have one of the members of our Cerulia board, Associate Professor Tina Soulis, with us, and again Hugh Taylor. And I'd also like to welcome some other very important founding members of our CERA community. That's Associate Professor Julian Wright, and also Associate Professor Justin O'Day and Sally O'Day, and Professor Noel Alpins, another long-term ambassador and supporter of CERA. And thank you also to our valued philanthropic supporters who are here tonight. There's a great number of you here in the audience, and we remain extremely grateful for your ongoing support. And of course, most importantly, we've got a lot of representatives of the Crock family up the front here, and this is we're really here tonight to honour Professor Gerard Crock's legacy of innovation in eye research, and so it's great to have so many representatives of the family here. So for those of you who are newer to the CERA community, I'll share just a little bit of history. So it was 1963 when Gerard Crock was appointed the University of Melbourne's first Ringland Anderson Professor of Ophthalmology at the ripe old age of 34. And his appointment was the first medical specialty chair of any discipline in Australia, and only the second chair of ophthalmology in the British Commonwealth at the time. Professor Crock really established the University Department of Ophthalmology and was responsible for a lot of the setup of the ophthalmology services at the Eye and Ear Hospital. And he clearly had a real drive to discover new ways to save sight. That's left a strong legacy behind for generations of eye researchers in Melbourne since then. And every year we come together to to celebrate that, and this event is now in its 17th year. And through his career, Gerard Crock saved the sight of thousands of people with eye disease through his pioneering surgical techniques. And so I think it's fitting tonight that we've got another pioneer as our speaker tonight, and that's Professor Pete Williams, who's dedicated his career to developing treatments for blindness due to glaucoma, which is also my interest. And Pete tries to understand what we do to treat glaucoma if lowering the pressure in the eye is not enough. And so he's figuring out what the mechanisms of damage are, how to protect the optic nerve from ongoing damage, and really how to repair and restore sight in glaucoma. And Pete's joined us recently in January from the Karolinska Institutet in Sweden, where he was head of glaucoma research and research group leader at St. Erik's Eye Hospital in Stockholm. And for those of you who don't know, the Karolinska Institute claims to be Europe's premier medical research university. Cambridge would beg to differ, but we'll have that argument at a different time. Yeah, right. Yes, since we left the EU, he claims we're no longer eligible in Cambridge. Yeah. So loads of research experience, originally from Cardiff in Wales, and then I spent a lot of time in the US and in Singapore and in Sweden, and has joined us as the lead of our Neuroprotection and Repair Unit. And he will run dual research programs in Melbourne and also continuing at the Karolinska in Stockholm. And I think that's a great way of demonstrating the global nature of the research that we do and building these partnerships and also bringing great expertise to Australia. So Pete has really been successful in his career so far. He's pulled in more than $20 million of competitive research funding. He's published in some of the leading journals in science, let alone ophthalmology, including Science, the journal, and Nature Medicine, Nature Communications, Proceedings of the National Academy of Sciences of the US. Holds 4 patents, founded 2 startups, including MIM Neuroscience, which he's bringing to Melbourne, has brought to Melbourne, and is an active part of 4 different clinical trials, including a trial of vitamin B3 in glaucoma, which I know some of you in the audience are actually part of, and which is probably the biggest trial of neuroprotection or series of trials of neuroprotection in glaucoma that has ever been held worldwide. And as if that's not enough, we've managed to rope Pete into doing the Lions Ride for Sight this year, which is a 320-kilometre ride through the less flat parts of Gippsland, which is coming up in a couple of weeks' time. And I'm doing as well. I'm cheating because I'm using an e-bike, but Pete's doing the whole thing on a conventional bike. So to raise money for some of the vision research that we do. And I think Pete really exemplifies CERA's approach to translation and getting the research out of the labs and into the clinic, and doing so with enthusiasm and vigour, as I think you'll see. So it's now my great pleasure to invite Professor Pete Williams to deliver the 17th Annual Crock Lecture. Pete.
Keith, that was very kind. You certainly earned a place on my Christmas card list. We have to go through that again. See? We'll get there. There we go. That's me. All right. Super duper. All right. So before I start, I want to stage why I'm here, right? And I think I want to get 2 important points across. And the first is that I see scientists on the spectrum of problem solvers on one side to people helpers on the other. So on one side, you've got your basic scientists. They're trying to do stuff in cells in the dish, in the lab. And on the other side, you have your clinicians actively helping people day after day. And my aim has always been to be as close to this side, the clinician side, as possible, but without stepping into the clinic myself. So that's the kind of niche I've carved out. And the other point I want to get across is, you probably tell I'm not a very anxious person, but I have one work-related anxiety. And that's that I'm going to end my life, and my tombstone is going to read, "Here lies Prof. Williams, 65,000 citations, 200 papers," and well, that hasn't really helped anybody. I don't want to be a content producer for Springer Nature. That's not something that's going to bring me joy. What I really want to do is push implementation and translation to the clinic. And so that's really why I've joined CERA, because that gives me the opportunity to do exactly what I love doing, which is taking something from the bench to the bedside, which is what I've named my talk. Right. I always start by thanking my team. I often get the opportunity to speak in really nice places, so I always joke that I leave everybody in cold, wet, and rainy Sweden, and I get to travel the world and present their research. But the reality is, it's a team that does all this work with me and for me. And the best way to describe that is, I'm a visual neuroscientist by training. I don't need a team of 20 visual neuroscientists. I don't need 20 clones of myself. That's not going to help me cure disease. What I actually need is a really multifaceted team of everything from, clinicians, pharmacologists, chemists, basic biologists, neurobiologists that can come together to really help me decipher disease mechanism and cure disease. We don't do that in isolation. This is just a little world map I put together. This is all the open grants I have right now with research funding projects for those of you not in the academic world. And what I just want to try and show you here is the kind of the international reach of the stuff that we're doing. And we've got this nice hub in southern Australia here, which is where we are right now, which is just the most perfect place to be, both for my family but for my science as well. So this is glaucoma. I just want to get a few points across before we start. So glaucoma is we often think of glaucoma as just a disease where cells are dead and then you're blind. And really, we need to remember that it is a chronic progressive dysfunction followed by the loss of the retinal ganglion cells. These are the cells that live in the inner retina, and their axons or cables exit the eye and become the optic nerve and go to the brain. And it's actually the death of these axons that is our clinical definition of glaucoma, that leads to our irreversible vision loss. It's a very complex disease. It's about 300,000 patients in Australia, about 80 million in the world, probably underdiagnosed, something more like 114 to 120 million patients worldwide. And unfortunately, depending on the demographic, between 10 to 40% of those patients are going to continue to blind us even if they have conventional glaucoma treatment. There's three major risk factors. This is age. The majority of our glaucoma patients are older. Genetics, monogenetic glaucoma is quite rare. So this is glaucoma where it's a single gene mutation. It's much more polygenic disease. I think we now have 129 genes that are associated with glaucoma through GWAS studies. So very much like Alzheimer's disease, where we talk about hereditary risk, but it's very complex. And in about 50 to 60% of patients, it's high intraocular pressure as well. And he's saying to me, okay, Pete, I'm here for a glaucoma lecture. I know all this. Why are you telling me this? So I put a little box around it. The important thing is we can't really change this very much in our patients, right? We can't change your age. We can't change your genetics, at least legally. And high intraocular pressure, treatment for high intraocular pressure only works in a subset of patients. But what we can do is we can model this in animal models of disease. And that's what gives basic research it's such a strong importance in ophthalmology. Something fun for you all to think of now that you've had a glass of wine in your system is how we all have to die. And cue laughter. And if we're lucky, we all get to the end of our life with a normal, slow progression of vision loss. So after about the age of 40, we're going to lose a small percentage of our retinal ganglion cells every single year. That's the grey dotted line at the top. Remember this slide. It's going to be important for the questions at the end of the show. And if we're lucky, we're going to end our life with pretty much normal vision loss, but our brain is going to do a really good job at mapping in those differences so we don't notice it. Unfortunately, in the case of what we can call a fast progression of vision loss, comparative to death, something like glaucoma, we have, a relatively large period of time for a lot of patients where there is dysfunctional or absent vision. So what we've really wanted to understand in my group is how we can identify the mechanisms of disease throughout the time course of glaucomatous disease. And if we can do that, we can then design therapies that target these and intersect. Hello. Come in, come join us. Okay. It's like a game show. Muddy Hall, what's behind door number 3? Hey, Mandy. Okay, very good. All right. Come in. Yeah. No problems. Very good. Good. So, if we can understand the mechanisms of the disease, we can start to design treatments and therapies that are valuable for patients across the disease spectrum. So if we can understand what happens early in the disease, we can design early interventional strategies. This is the blue dotted line. And hopefully that means that we can intersect disease relatively early on and give patients relatively good vision throughout the rest of their life. But even our patients that have lost a significant portion of their vision, if we can identify what happens later in the disease, we can extend that period of functional vision. And it might only need to be a couple of points in their visual field or a couple of decibels, but it makes a huge difference if you can increase their working vision by 3, 5, 10 years. This is more time to read the newspaper, have your driver's license, which we often say is, the key thing that people won't play with your grandkids, whatever. So that's what gives me the motivation to do this, is to extend this period of functional vision. So what I've done then is I've kind of formulated my research into 3 research pillars. In my first pillar, I want to understand what I can do for glaucoma patients now, today. There's no good me designing something that's no good for another 100 years, but what can I do to help people today? That's going to be the first third of my talk. And the second pillar is, how do we take that information that we've learned and design a drug therapy that is regulated? So something that you can go to your doctor directly and have that prescribed and billed. And at the end of this, I want to understand, can we actually make a one-shot therapy for glaucoma? So something that we can give to patients at different stages of the disease and halt their neurodegeneration, or even potentially restore lost vision. And I think this is my immediate goal, my 5-year goal, and my 10 to 20-year goal. And I think we're on track. We're going to do this slide at the end. We're going to talk about how far we got. So to do that, I like to think about the aging retinal ganglion cell as an old motorcycle trying to climb up a hill. So right now, when we treat glaucoma, we look at the retinal ganglion cell. We focus on factors extraneous to the motorcycle, okay? We try and lower the hill. That's lowering IOP. We try to improve the road surface. So these are surgical devices, the way we run our clinical trials. And sometimes we just stand there and we go, oh, why is the hill so steep? I don't want to cycle up this without my e-bike. And that's our genomic studies. And these are all really, really, really important. And they're very valid clinical strategies. But what you can already tell me is that none of these strategies target the motorcycle directly. We could bulldoze that hill flat, and if the motorcycle is still broken, it's still not going anywhere. So what we want to do is focus on the factors intrinsic to the motorcycle, intrinsic to the retinal ganglion cells. Can we improve its engine efficiency? Can we add a turbocharger? Can we give it premium gas? If we target all these factors together, we can allow the motorcycle to climb the hill, regardless of how bad it is, allow the retinal ganglion cell to climb the hill of high intraocular pressure. And most importantly, to the clinicians in the room, I'm not trying to take your jobs. You can keep doing your surgeries. These are all going to be complementary to the strategies that we already have in the clinic. So what can we do for glaucoma patients now? Okay, so these studies started off lots of research in animal models. And to cut a really long story short, we were interested to understand what happens to the retinal ganglion cell at very early stages of disease. So this is at a time point where intraocular pressure is high, eye pressure is high, but there's no detectable neurodegeneration. We used a variety of different tools, including RNA sequencing, transcriptomics, looking at how genes change in the retinal ganglion cells. And we identified really marked changes to gene expression signatures for genes encoding mitochondrial and metabolic processes. And if we drill down into this in much greater detail with electron microscopy, we actually identify kind of a loss of mitochondrial integrity at the level of the synapse and the dendrites. So this is where these cells are connecting to other cells in the retina. You're saying, Pete, that's all well and good, but I don't really care about clearing glaucoma in mice, and I think that's a very, very valid question to ask me. And so we teamed up with Mike Fouch at the Mayo Clinic to do a really beautiful experiment using human tissue. This is from Mike had developed this great resource of human tissue where the donor eyes, the glaucoma donor eyes, had been taken at a time point where they had visual field examinations done quite close to the time of death. That means that we could look at this tissue and go, we can predict pretty closely what the retina was like at the now in the dish versus at the patient's last appointment. And we use something called serial block face scanning electron microscopy, or SBF-SEM to its friends, which is a way to reconstruct tissue at very high detail in 3 dimensions and start to look at what's happening to the cells. And to, again, long story short, what we identify in our human glaucoma patients is exactly what we saw in the mice as well, which really validates our approach. We've gone on to further try and confirm this in a large biobank of human glaucoma tissue we have at St. Erik's Eye Hospital, which is one of the largest eye tissue banks in the world, where we see the same thing again, which is a loss of mitochondrial content in glaucomatous patients. One of the most important findings from these series of experiments was the discovery of an age-related decline in retinal NAD, which has now somehow got typoed on the slide, so my apologies. NAD is a really important redox cofactor. It's a very important metabolite, and it has a very well-known role in neurodegeneration. NAD is made in your cells by three routes. Through de novo, so from your diet, from tryptophan, amino acid you get in your diet, through the Price handler pathway, this is nicotinic acid or vitamin B3, and recycled through nicotinamide, which is another form of vitamin B3. Neurons only have the capacity to make NAD through the salvage pathway. This is why it's the only one I'm showing you on the board. It's a very simple two-step reaction that goes from nicotinamide or NAM to NMN to NAD and then recycled back again. So I led with my working hypothesis then is that actually it's declining NAD, which is this really critical metabolic insult during glaucoma that drives glaucoma pathogenesis with age. And so I'm a very simple person. I really only like experiments where I can take something away or put something back in again. And that's exactly what we did, which is we took nicotinamide, so this form of, not the keyboard, this form of vitamin B3, gave it at oral doses to various different animal models of glaucoma and see if we could prevent the neurodegeneration. To sum up many, many years of research into one slide, which is really, really sad sometimes, I get the point across that in our initial experiments using one particular mouse model of glaucoma, we had an almost complete neuroprotection with oral vitamin B3 or nicotinamide. We confirmed this in 6 or 7 more animal models of glaucoma and optic neuropathy. We've now had 3 groups independently validate this across the world. So I think it's probably one of the most reproducible findings in a glaucoma animal model. And as we drill down into the mechanism of this, everything we look at that could change in the retinal ganglion cell, whether it's metabolic function, it's synaptic connectivity, it's electrical firing, it's connection to the brain, everything is fixed by preventing neurodegeneration with high NAD. So nicotinamide then has this really amazing potential as a neuroprotective therapy because it's very safe. We've got about 80 years of clinical history on nicotinamide. It's very well tolerated. You can take it in grams a day, even in children, and it's very affordable. So the health regulatory agencies say that anything that's made with corn has to be fortified with nicotinamide because corn is tryptophan deficient. So if you look at your breakfast cereals, your cereal bars, your power bars, your energy drinks, they all have niacin or nicotinamide in to fortify. So it's very, very cheap to produce as well. So that's great, but I'm just curing mice. I want to do something a bit better than that. So it's really my pleasure to be part of the very first clinical trial for nicotinamide and glaucoma, which was run from CERA by Johnny Krausen and Flora, who sat right in front of me, where we performed a crossover experiment. So this is just half our patients took the placebo, half took nicotinamide. They crossed over and then had the other treatment. This gives us a really great way to power a clinical trial, with the endpoint being something called the photopic negative response of the ERG. So this is an electrical waveform of the retinal ganglion cell that tells us about its health. After quite a short treatment, just a few months with nicotinamide, we had a really marked improvement on visual function in existing glaucoma patients on nicotinamide, with over a quarter of them actually improving on their visual function, and very high adherence rates on nicotinamide as well. I like to joke with Flora, we have a lot of patients. You need to remember that our glaucoma patients are predominantly elderly and predominantly female. And one of the positive side effects of nicotinamide is increased hair and nail growth. So we quite often get these very funny phone calls or emails to say, "Oh, I'm really worried I've unmasked myself from the trial because I've been really good at concentrating, and my hair and my nails and my skin have improved so much." And I like to joke that even if the clinical trials fail, I've helped 500 old ladies feel good about themselves. So that can be my contribution to science, if nothing else. So we've continued to drill down into exactly what's happening in glaucoma patients and whether this is something really relevant to them. This is a really beautiful paper we did with Ted Galway-Heath at Moorfields Eye Clinic, we published last year, where we took glaucoma patient blood cells and actually show that their capacity for energy metabolism and their capacity for NAD is lower systemically. So that's very important. So this is something that is innate to glaucoma patients that is making them susceptible to high intraocular pressure and further neurodegeneration, as opposed to something that's happening very locally in the eye itself. So it really helps validate our approach. Nicotinamide is really great in the animal models. It's good in the human patients, but it doesn't fix everything for everybody. So we really need to understand what else is happening. So we came back to our mice again. You're going to see me hopping around from mouse to man quite frequently. And for this, I really wanted to understand what was nicotinamide not doing. So to understand that, we took glaucoma-prone mice and rats, treated them with nicotinamide, and then performed a multi-omic profile. So this is transcriptomics of genes, metabolomics, metabolites, and phosphoproteomics, so the way proteins are phosphorylated or changed after they're made. And what we identified was a metabolic nexus controlled by NAD and pyruvate that leads to a metabolic block in retinal ganglion cells. If we overcome that metabolic block using nicotinamide, we've already done that. Using pyruvate, this is a simple sugar. Bodybuilders use it, for example, or with rapamycin to reprogram cells. We could actually get a phenomenal neuroprotection. Again, I don't trust if I've only done this myself, so we shipped off the methods with Simon John at the JACS. We repeated it in 2 different universities in the US and with Bob Caston's group in Adelaide as well. We all got the same results. So that made me very confident enough that we could then do the next clinical trial, which was the combination of nicotinamide and pyruvate together. A very similar paradigm to before, looking at visual function after a relatively short window of treatment. And we see the same thing again, which is an improvement in visual function in glaucoma patients taking the nicotinamide and pyruvate treatment. So this was run out of Hawkins Eye Institute, so Columbia Medical Center, New York, with Jeff Liebman and Gusta Morales. Very good. So what I've hopefully convinced you of in the last 6 or 8 minutes or something is that glaucoma is a disease that has a very strong metabolic component. And if we can correct that, we can actually provide a very strong neuroprotection in animal models, but we can actually start to improve visual function in glaucoma patients as well. So I often like to say, maybe to the chagrin of ophthalmologists, I actually think glaucoma can be a treatable optic neuropathy. We've done all the valid animal and preclinical experiments that validate a lot of the work that we've done, but now we really need the RCT, so the controlled clinical trials, to really assess this so we can take that into the public domain and give that to the clinicians. And so that's exactly what we're doing. This is called TGNT, or the Glaucoma Nicotinamide Trial. 2 sites here in Australia, CERA being one of them, 2 in Singapore, 6 in Sweden, I think 11 hospitals in the UK. This is just a little overview of where we are. We're about 2/3 enrolled, and we're looking for a completion in 2028 currently. So this will be, at its completion, the largest neuroprotective clinical trial we've ever done in glaucoma. So that's very exciting as well. So if it doesn't work, it's not because the trial failed, it's because nicotinamide doesn't work. So we'll know the answer one way or the other. So I think that's very exciting. And it's great to be part of these big international leadership teams really trying to change the way that we treat glaucoma. We've also done a lot of work on what's called non-energy metabolism, so the way that we form the different building blocks and amino acids in our cells. One thing we identified in both animal models and glaucoma patients is dysfunctional 1-carbon metabolism. This is a really important way that we regulate DNA and we build amino acids in our body. We teamed up with, again, colleagues here, colleagues in Melbourne and Moorfields Eye Hospital to really decipher what is going on both in animal models and glaucoma patients, and identified the methionine cycle, so the way we make methyl groups of DNA, as a central nexus for how the damage occurs after the initial intraocular pressure insult. So importantly, this is now a retina-specific insult driven by high intraocular pressure. So it's very different to the nicotinamide and NAD story, which is a systemic effect which susceptibilizes glaucoma patients. So this is only valid for the ocular hypertensives. We developed a really nice treatment overcoming this cycle. So there's 4 entry paths into the methionine cycle: vitamin B6, B9, B12, and choline. And if you supplement one of those, you get a small protective effect. If you do all 4 of them, you get a very strong protective effect as well. And we performed we did a new Mendelian randomization analysis to look at genetics of glaucoma patients. We also did dietary recall through the UK Biobank. So this is looking at hundreds of food diary entries for hundreds of thousands of patients. And what we actually identified is that patients that have a higher level of this in their diet actually have a thicker RNFL, so thicker axons in the retina as well. So that has led us then to directly do our phase 2 clinical trial. We're doing this in Sweden. This is the unfortunately terribly named IVMGS. I have to read that out loud because it's so hard to remember. Interventional vitamin mixed glaucoma study, an open label study with about 100 glaucoma patients. Doing this combo B6, B9, B12, and choline, and that's finished recruiting. We should have the data by the end of this year, early next year. So that's exciting as well, because again, all these things are in the public domain and available over counter. In the second pillar, then, great. You said, Pete, okay, that's cool, but that's just a lot of vitamin therapy for glaucoma. How can I get my doctor to actually prescribe something for me? We're going to come back to NAD, so you all remember this pathway from before. Again, it's on your quiz sheet. What I've taught you so far is that low NAD, or we should really say low NAD generating capacity, susceptibilizes the retina to neurodegeneration. If we restore NAD, genetically or pharmacologically, we can provide a very strong neuroprotection, and we can start to show protection in glaucoma patients as well. So now what we have to ask is there a way that we can make a retinal ganglion cell, a retina-specific therapy for NAD? Right now, high levels of vitamin B3 orally is doing a lot to the body. It's not very specific to the retina. But we have this beautiful pathway, this NAD salvage pathway I showed you before, nicotinamide, one-step reaction to NMN through NAMPT. We don't touch NAMPT, that's a cancer target. And then NMN to NAD. NMN to NAD is through two enzymes, NMNAT1 and 2. NMNAT1 is in every single cell in the body except for red blood cells, and NMNAT2 is only in neurons. So already we have a potential neuron-specific target. Working with Ray Wong colleagues here at CERA, this is single-cell sequencing from human retinas and beautiful histopathology from my own group in Sweden, just to show you that not only is NMN restricted to neurons in the retina, it's almost completely restricted to retinal ganglion cells as well. So if we can design something that targets NMNAT2, we can make an NAD-specific therapy almost specifically for retinal ganglion cells in the retina. So that gives us a really good target. So that's exactly what we've been doing. NMNAT2 itself actually declines in the retina in various different animal models of degeneration, but also in human glaucoma patients as well. So we know that it's doing something. Importantly, when I say it declines, it's not absent. If it's absent, you'd be dead. It's very essential for life. But we see this as well in both Parkinson's, Huntington's, and Alzheimer's disease patients as well, about a 25 to 50% decline in this protein during disease. So we can come back to this pathway that you all remember, and I'm going to refine my hypothesis now that it's actually declining NMNAT2 that's the critical risk factor for glaucoma, not just NAD. And again, I'm a creature of simpleness. I really like experiments where we put stuff in or take stuff out, and so that's exactly what we've been doing. We've been asking the questions, what does NMNAT2 do? Do high levels of the gene or the protein or its activation state drive protection of the retina? And do low levels of this, either genetically or its activity levels, drive neurodegeneration? We've done all those experiments in various different iterations. What I'm going to tell you about now is the discovery of a really potent, novel neuronal NMNAT2 activator called EGCG, epigallocatchin gallate. This is one of the major components of the green tea polyphenols. It does a really good job of driving NAD in neurons. So that's retinal ganglion cells and cortical neurons. That's what we test in. If you've got another favorite neuron, I don't know the answer for you. But it does so a lot better, 1,000-fold better than nicotinamide does. So that's great. And it also prevents degeneration to the retinal ganglion cells in the micromolar range, so very, very low rates. And you're saying, Pete, that's well and good, but now are you peddling me Chinese herbal medicine? I say, well, we've done all the correct experiments to show you that we do have a mechanistic pathway for it by taking the genes in and out and changing the pharmacology. And you're saying, okay, Pete, great, Chinese herbal medicine in mice, how is it relevant to me? Well, then we can also do this in patients. So this is explants of human retinas from donors from the Cornea Biobank. And what I want you to appreciate is when they come fresh out of the eye, they look like where it says day 0, so that's days ex vivo, so days after transplantation, top left corner, lots of white dots and lots of retinal ganglion cells. After 7 days, they're all dead. But if you feed them nicotinamide or EGCG, you get a very strong protection of human retinal ganglion cells as well. So that's nice evidence that we might be going in the right direction. So that gives us a really novel opportunity then, which is we can use EGCG to be what we call a tool compound to actually develop new drugs. Okay, EGCG itself is a very poor, chemically a very poor molecule, but we can use that as a backbone or a scaffold to design new drugs. That's exactly what we do. So this is just an oversimplification, even though it's hard for me to understand. When we take EGCG, we do what's called a structural simplification. So that's we break down its structure until we find the moieties of that chemical that are important for what it's doing. We identified this core, and then we broke that down and identified this tetrahydroquinoxylene core. Don't ask me to say that 3 times with a glass of wine, which we've used as a backbone then for developing novel drugs. This is what that looks like. Through different iterations of medicinal chemistry, we make drugs that make more and more NAD. This is, again, in brain neurons. As our proof of concept, every time we go through these different iterations of chemistry, we get better and better drugs. And what does that translate to? If we go back to our mouse and our retinal injury again, it equals better and better neuroprotection. So now we have started to develop drugs that are NMNAT2 specific, so retinal ganglion cell specific in the retina that save retinal ganglion cells. That's quite cool. So where are we now? As Keith mentioned, we made a company of this 6 years ago. Last year I moved that to Australia as well. We're in our 9th, 10th, and 11th iteration of drug synthesis. We've developed about 130, 135 novel compounds that we've taken through various different rounds of what's called ADME tox, so the way we test drugs to see if they're doing what they should be doing. And now we're preparing to test in vivo as well. So our aim here is that we would like to make actually an injectable therapy for glaucoma, so something we could put in the eye and arrest neurodegeneration, but also we're following up oral formulations as well, so we can make the tablet form. So lastly then, before I get the shepherd's crook pulled off the side of the stage, is can we develop a long-term protective or regenerative gene therapy for glaucoma? So we're going to come back to my good friend NMNAT2 for the purpose of this exercise. We can introduce that via a gene therapy. So this is adding in a construct for a single injection into the eye to increase the levels of NMNAT2. If we do that under the effect of a clean exotomy, so this is severing the optic nerve, we get a very strong neuroprotection. That's because we're targeting that pathway. It's basically a 100% protection of the ganglion cell. If we take mice and we remove the mouse version of NMNAT2 and put the human version back in, it works as well. So that means it can overcome these stresses. And if we go into our chronic models of ocular hypertension, so our models of high intraocular pressure, we also get a nice neuroprotection as well, remembering that this is a gene therapy that specifically selects retinal ganglion cells. We're only looking at saving the retinal ganglion cells and their axons here. So that's great, that's neuroprotection, but can we go one step further and start actually regenerating the optic nerve as well? So this is work that I'm doing with Richard Ever at King's. This is just the animal model, you don't need to see that. But basically what we've been doing is designing a lot of dual vector gene therapy. So this is where we're putting 2 or more genes into a single gene therapy vector, putting it in the eye and seeing if we can drive both protection and regeneration. One of these molecules is called protrudin, which Keith was also part of the original studies. Protrudin is so-called protrudin because it makes protrusions in cells. Us biologists are very exciting people. And the good news is that if you put too much protrudin into a retinal ganglion cell, you can make it regrow its axon very nicely. So what we get then, I'm just going to walk you through the pretty pictures at the bottom because that's actually what you want to see. So the first on the left that says NTGFP, this is what a normal retina looks like. This is in the rat. The purple round blobs are retinal ganglion cells. The green lines are axons, so you can see lots of cells, lots of axons. Good. Following ocular hypertension, that's the next box, OHDGFP. Following ocular hypertension or glaucoma, there's not really anything left. If we treat it just with NMNAT2, that's the box in the middle, we get lots of cells, but not very many axons. If we treat with just protrudin, we get lots of axons and not very many cells. And if we treat with both together, which is the one at the end, it looks almost identical to the first slide, which is nice. So now we have a nice way to increase retinal ganglion cell survival. So the logic here is that if you really want to do successful regeneration of the optic nerve, you have to provide a protective environment for that to happen. So that's what we're doing. No more neurodegeneration studies. This is just a nice little slide with some cortical neurons in a dish. You can grow them, they'll all overlap, the little filaments on top of each other. You can scratch them with a scalpel blade, and they'll regrow into that space. If you use the gene therapy for either protrudin or the dual gene therapy for protrudin and NMN82, you get more regenerating axons, and we can also get regenerating axons down the optic nerve as well. So this is an injured optic nerve, and the white lines, that doesn't come up very well on the board, I'm very sorry, but please believe me, those are axons growing back down the optic nerve trying to make their way to the brain. How do we take this into the real world? That's all well and good, me showing you stuff in addition in a rat. We're just about to form a spin-out company, which Keith is also part of, called Ira Bio. We just received a fairly sizable VC fund to start this as a company and start developing this with a milestone to get to the IND. So this is the filing in preparation for a clinical trial. The last thing I want to show you is work that I've been doing with Rick Liu here at CERA, where we've been utilizing the switchable gene therapy approach. So it looks very complicated, but it's actually conceptually very simple. In the case of a monogenic disease, so a disease caused by one gene, you can do a gene therapy and you can change that gene or correct the mutation or put the gene back in, and nothing bad is probably going to happen because it was already dysfunctional. If you're starting to talk about a gene that's maybe involved with a protective or regenerative pathway that shouldn't be in that cell in the first place, then there's a much larger regulatory hurdle that you have to come over. So we have to convince the regulators and the patients we're going to treat with this that we have a way to control it. One way to control that is to make sure that these proteins are made on demand. One way to do that is through using destabilizing domains that have been identified either in bacteria, that's a DHFR on your left-hand side, or FKBP, which is the mammalian destabilizing domain. What this does is you stick that on the front of your protein, and when it's made, it's degraded. And you think, well, Pete, that's not very useful to have a protein that doesn't do anything. But if you give it a synthetic agent that stabilizes this, in this case, the bacterial domain is TMP, a common antibiotic, or for FKBP, this is called SHIELD1. This is a rapamycin analog. You can stabilize the protein for about 24 to 48 hours after administration. That means that you can basically have these proteins stable and active on demand. And the way that Rick is doing this, which is really elegant, is doing this via eye drops. So therefore, the principle here would be is you have your singular injection for glaucoma, and you take an eye drop that actually keeps that protein on or off when you need it. So what we've been doing then is this is what it looks like in the retina, just making retinal ganglion cells go different colors if we turn them on or off. And we can do that with our favorite gene, just as a proof of concept, NMN82. And all I want you to appreciate from this is in the top row, there's not a lot of color, and in the bottom row there is a lot of color because we've turned it all back on again. That's the simplest way I can make this slide. The aim here then is that we could actually make a single therapy for glaucoma patients whereby at the point of diagnosis we give you the injection that has both the protective and the regenerative gene therapy in. You take the eye drop for the protection until you start to lose vision, then you can take the eye drop for the regeneration when you need it. And that overcomes probably a lot of regulatory hurdles to do with regenerative gene therapy as well. So that's it. You'll remember the pillars. So I'm going to ask the question, where are we now? This is the quiz at the end. So pillar 1. We've got to complete the nicotinamide trials. I think we're very close. We're now, really 2.5 years away from having the real clinical answer and actually informing the clinic about this. We've got lots of other different trials that we're doing, and Flora and I have got other stress testing to do with nicotinamide that's ongoing as well. So I think the chance of us having something in a patient's hands, the glaucoma patient right now, we're there. So that's good. Tick. We're at a point with the NMN82 targeted drugs that we're actually doing our safety and efficacy studies. I think we're probably 2 to 5 years away from doing our first clinical trials with these. We've actually got a CRO, so contracted research organization now, doing the safety studies of this in vitro in mice for us. So we're one step closer. And I think we are now genuinely one step closer to developing a really protective and restorative one-shot gene therapy for glaucoma. So I told you I wanted to do something now, in 5 years, and in 10 to 20 years, and I think we're on track for that. And that happens because people do stuff for me and and help me, so I don't have to do it all by myself. So that's really who I'm grateful for. And that's it for me. Thank you so much.
Pete, that was absolutely a wonderful talk, and I don't know how much nicotinamide you take yourself, but there's clearly
I'm 55.
there's clearly a lot of energy in there in terms of the work that you do. And Pete, before he even arrived at CERA, I think you had collaborations with every single discovery researcher at CERA before you even hit the ground here that have been built up over the years. So you can see that we reckon he's a pretty good fit for the work that we're doing, and incredibly collaborative and doing some great science. And thank you for walking us through some of the really exciting things that you're doing now and are planning to do. So that was really fantastic. So just before we open things up to the floor, I'm going to introduce some new conversations. Pete's sat up at the front here, but I'd like to introduce, first of all, Lani Din. So Lani is a blind woman who is living with glaucoma currently, and she's an advocate for accessibility and inclusion for people with low vision. She's also a very experienced administrator and operations professional working for a major hotel chain and previously worked in the travel industry in Vietnam. She's also passionate about community engagement and inclusion and education and awareness and connection. And also a keen sports person, so she plays basketball and has been part of Tennis Australia's program for blind and low vision people in that sport. And as if that weren't enough, I think, Lani, you're currently learning to skydive as well, which is, amazing. I tried that and couldn't give it up fast enough, I tell you. So welcome, Lani, and please come and join us. And then rounding out our discussion tonight is Dr. Flora Hui. So Flora is a glaucoma researcher at CERA. She's been seen on the screen several times tonight because she's part of many of the projects that we're doing here. She's an optometrist and a clinician scientist who's been involved with the vitamin B3 work right from the very beginning and is leading the clinical trial here at CERA. She's also part of the NOMAD trial, which is another trial using a vitamin B3 to try and improve retinal function after retinal detachment surgery. And she's a fantastic science communicator and promoter of STEM education and careers. She's a graduate of the Superstars of STEM program and is also selected for an internship at the ABC as part of their top 5 program as well. So Flora, welcome, and thank you to all of our panelists, and I'm going to hand over to you.
Hi everyone, and thank you Keith, and congratulations Pete. That was a fantastic lecture. I admittedly have known Pete for about 10 years now, so I do know the story, and I have worked with him quite intensely over the last 10 years, if it wasn't apparent.
Intensely being the key word.
And we continue to, and back then he was actually still a postdoctoral fellow in the United States. That's when I first met him at a meeting in Austria. So over the last 10 years, it's been a pleasure to see how the work has evolved and amazing to see all the progress. And it's a privilege to be part of the 3 pillars to try and help make this a reality. And I also want to flag that, some of that work that we did here at CERA, we're taking something from the bench to the bedside, as Pete says, was only made possible because of philanthropy, because our clinical trial to do something first in people is a bit blue sky and actually required philanthropic donations to make it possible. So it's fantastic that we had that opportunity to take that clinical trial. And we actually took that from, him publishing it in 2017. We published the first clinical trial in 2020. So it's 3 years, which is an amazingly short time to be able to do something like that. So that's fantastic to see. And Lani, thank you so much for joining us. It's really wonderful to have you here today. And I'd actually like to start off with you because, many people here know about glaucoma, but I'd really like to hear a little bit about your story about, when you were how you were first diagnosed with glaucoma and how it's actually impacted your life. So I'll just pass it to you.
Good evening, everyone. My name is Lani. I didn't lose my vision in one time dynamic moment. It happened very slowly, and my eyes became very sensitive to light, and they watered all the time. and everything started to look cloudy. At that time, it was in my early 30s, and no one thought it could be glaucoma. And when I was finally diagnosed, the damage were already quite serious. And at that moment, I realized I was not only losing my vision, I was also losing my confidence. I kept asking myself, how am I raising my child? And if I cannot see clearly, how can I keep working? And how can I still be me? There were years when I felt very lonely. And during COVID, my mental health getting very difficult. As a blind mother raising the two boys, I often ask myself very simple question: am I enough? But living with a glaucoma, it changed the way I see the life. It taught me patience, and it taught me to listen more carefully. And it taught me that some days my capacity is lower, and that is okay. I stopped asking why me instant, and I started asking, like, what can I do with a new experience? Yeah.
Thank you, Lani, and thank you for sharing. And I think it's really important to, especially a lot of us in the room are researchers here, to remind ourselves about why we do what we do. So I think it's very important, and thank you for sharing your story and your journey with us. And I'm really glad to see how it's also empowered you as well. I mean, skydiving, I have not tried skydiving, and I don't think I'll be doing that anytime soon. So I think it's fantastic that you are still taking life, living it to the max. And
because I got a dream like I'm blind, but I wish I can fly. That is my dream. And that's why that is serious my experience, because I already the blind and low vision tennis player, and I can play well in blind low vision basketball, and I play the blind golf, and now I really want to get the license for the skydiving.
Amazing. I'm going to have to keep in touch with you to see how you go with that, because that would be, I'd love to hear your stories about it.
Yes.
And so I'd love to take it back to research for a second.
Yep.
And, why do you think research is so important in glaucoma?
When you live with a condition that cannot be reversed, hope looks different. Hope is not always about getting your sight back. Sometimes hope means the earlier diagnosis and better treatment and better protection. And for me, research is not just science, and it is very personal. It's about the future. And today I live with a glaucoma, but I think I accept it, and I really hope in the future. And I still have so many dreams.
And I'm sure you can achieve them all. Thank you.
Yeah.
Thank you, Lani. And that's actually a really lovely segue into Pete, actually. And Pete, I might just ask you to grab the microphone because, your 3 pillars are amazing. And, I think it's actually a really exciting time to be in glaucoma research right now. So my question to you is, what do you think is the next big thing?
Yeah, that's a good question. So I often maybe cruelly joke that ophthalmology is 10 years behind cancer and Alzheimer's disease. And the reason I say that is that we watched all these huge kind of moonshot initiatives over the last 15 or 20 years that's really kind of changed how people talk about cancer and Alzheimer's disease, using those two as an example. And we are at that point now for a lot of optic nerve and ophthalmic diseases where we've had these amazing breakthroughs that have, I think, really stemmed from the inherited retinal diseases where we've had the monogenic gene therapies, this Luxturna and the single, $1 million injection that can cure blindness. And that's really given, I think, the ophthalmology field just a huge prod that it needed for innovation. In the background, there's been a lot of really exciting underlying technology that's now being utilized by us in ophthalmology. That's really speeding up the way we do stuff. That's not only the way that we talk about and see patients, but also the way we integrate big data, how we utilize healthcare data sets across countries, which really help us define the disease better as well. On top of that, we're at a turning point for drug therapies. I think neuroprotection is getting more and more validated as a strategy for glaucoma. Gene therapies I've mentioned are on the ball. Stem cell replacement therapies I think are going to become a thing for optic nerve disease sooner rather than later. We've had this already, some success in outer retinal disease, and now I think inner retinal disease is next. I'm actually, there's a large, if anybody's interested, there's a large consortium called the Restore Consortium, which is looking at how we perform retinal ganglion cell transplantation and optic nerve regeneration. That's 200 groups across the world that are together as a consortium that are trying to do this. And not only are we working together to really, fix some of these underlying, technological problems with stem cell replacement therapy and optic nerve regeneration, but we're also actively communicating with the FDA, because it's US based, about what do we actually need to do to define what a successful clinical trial for a glaucoma patient would be, and we don't have that right now. Of course, you've probably all seen in the news about whole eye transplantation as well, and that is, it's those kind of moonshot initiatives. If you ask me, Pete, am I going to have my whole eye transplant in the next 5 years? So you're not going to have it in the next 50 years, but it's important that we lay all this groundwork, because then we can build off it. I think what's important about everything I've just said is I haven't mentioned the word intraocular pressure.
But let's not forget about intraocular pressure, because as we know, a lot of people actually think about glaucoma and think about it as associated with having high eye pressure. And in actual fact, we used to think that glaucoma was caused by high eye pressure only. And so that's why all of our treatments are targeted towards that. Whether you're using eye drops or laser or surgery, it's all about lowering eye pressure in the eye. And a lot of this research has come about because we realize that eye pressure is not the only answer, but it's the only therapy that we have. So what else can we do? And I think it's actually a really exciting time, because we're starting to change the conversation a little bit, thinking about what else can we do in addition to lowering eye pressure. And that's what all this work today that Pete has shown you is about. And it's really exciting to see. But of course, we can't forget eye pressure. And so that actually, and of course, that's still a focus that we have here at CERA. And for example, we have some research looking at surgical devices. So a shout out to Professor Michael Coote, who I don't know if is here, but some of his team is here, who are looking at Vivid Flow, which is a clinical trial that's running here at CERA at the moment to look at, can we actually design a new surgical device that we can implant in people that actually helps lower eye pressure better and produce better outcomes for people with glaucoma. And so things like that are really important that we can improve upon, whilst also looking at alternative therapies and what else we can do with people with glaucoma.
Absolutely. And while we talk about clinical trials, I think maybe, Flora, you can comment on how running a clinical trial in glaucoma is actually becoming more and more feasible. We're no longer at a point where it's taking 10 years plus to run an effective clinical trial for glaucoma.
Yeah, and that's really interesting, and that's always been a sticking point in some of the glaucoma clinical trials, is that it's taken so many years to find an answer. And so some of the things that we try to do at CERA as well is how can we shorten the time frame to actually find answers earlier, so that we can find answers and deliver therapies into patients' hands and doctors' hands earlier. And as Pete mentioned in some of the in the vitamin B3 trial that we did for the first time, it was actually a 6-month trial. And that was, typically glaucoma trials take many, many years. And that's because we used a novel technique to try and look at, can we actually detect changes earlier in people with glaucoma? So some of our work is really focused on that too. So how can we make things faster and therefore also cheaper to run? Because clinical trials are also notoriously expensive to run.
But we have the advantage of cerulean and also the very favourable clinical trial design of Australia and New Zealand that really helps us expedite that locally as well.
Yes, and so thank you for bringing MIM to Australia, and you know, I'm a bit biased because I'm part of it as well, but you know, Australia is a great place to run clinical trials as well, so hopefully we can definitely explore some of these new therapies and bring them into patients' hands as soon as possible. And so I think that's enough talking from our part. I think it's time to open the floor to some questions, and so we do have some roving microphones around. We've got Matt and we've got Fleur, so feel free to put your hand up if you have a burning question, and Callum, if there's anyone online who has a question as well, then feel free to shout out.
Hi, I was part of the P3 trial.
Hi, Sam.
So you've sort of talked about IOP and sort of dismissed IOP, but is there any movement to producing an affordable home-based pressuring system? I mean, I measure my blood pressure with a little cuff. It's very simple. Is there anything on the horizon for measuring? Because you go and see your ophthalmologist every 6 months, and what the hell is happening in between that 6 months? Are you doing the drops properly? Are you controlling the pressures? What's happening? And I'm all for generating my own data to see what's actually going on.
Well, first off, thank you, Sam. So Sam's one of our lovely clinical trial participants, and I definitely did not pay him to ask this question. And but thank you for still supporting us and coming to our events even after completing the trial. But that's a very fantastic question, and home monitoring is actually a big area as well. And I didn't mention home monitoring, but there are devices where you can actually measure your eye pressure at home. But actually, some of the research done at CERA is about how can you measure your visual fields at home as well, because often you don't visit your optometrist or your ophthalmologist very often, so your vision might actually be actively changing. And the reason why it's actually quite hard to know if you have glaucoma is because glaucoma affects your side vision first. And so our side vision, we don't really naturally pay much attention to our side vision, and so if we're losing our side vision, we may not notice it. And it's tests like a visual field test, which actively tests our side vision, tells us whether things are actually getting worse. And so some of the work by Associate Professor George Kong, who's also part of the INEA, but also at CERA as a researcher, he's been developing a way to actually measure visual fields at home, and he's been really successful actually. He's been done, and he's got collaborations all around the world to actually look at the results that they get at home is comparable to what you get in the clinic. Yeah. But it means that you can actually measure it yourself, and the results get sent to your doctor. And if there are any reasons why they might be detecting early signs of progression that you might be losing vision, then essentially you get called in and can actually be seen by the doctor. So we are working on these kind of things as well at CERA to try and detect progression earlier, and that's a real active area of research that a lot of us at CERA are doing, including
and we've had
myself.
trials in this around the world now. So one of the ones we had recently in Sweden was upgrading now from having like the laptop screen with the camera to actually a pair of VR goggles where you can do this in situ. So you look funny doing it, but I think it's quite cool.
And I know you don't like visual fields, Sam, but they are important.
Flora, over here.
Oh, hello.
I have two questions, if it's okay,
Yes.
for Pete. Pete, my first question is there any suggestion that depressed nicotinamide might play a role in other neurodegenerations? And my second question is, given that you've had a global career now with diverse sources of funding, do you have any observations about the way research is funded in Australia now that you've been with us for a little while?
All right. Well, I'll take number one, and if I see a small red dot line up on my forehead during question number two, I know I'm saying the wrong answer. So question number one, do we know about low NAD and other diseases? The answer is yes. The mechanism by which axons die, programmed axon degeneration or Wallerian degeneration to use its posh term, is controlled by low NAD. If you take any neurodegenerative disease, whether it's Huntington's, Parkinson's, Alzheimer's, peripheral neuropathy, diabetic neuropathy, chemotherapy-induced neuropathy, it's due to low NAD. So this is being trialed in lots of different areas. There's lots of different NAD-related precursors and analogs that are being trialed. There's a really big market for this in the longevity supplements, which I'm trying to stay very far away from as well. That actually makes glaucoma a really beautiful tool disease because it's a very clean optic nerve degeneration, which if you go back 50 or 60 years, we used to crush the spinal cords of mice to test for axon degeneration, regeneration. Then they moved, that was a little bit barbaric. So that was, paralyzed the whole mouth. They moved on to what's called sciatic nerve transection. So cutting the sciatic nerve. Okay, well, that's just a leg. That's not too bad. And now actually the whole field has almost entirely moved to optic nerve, damaging the optic nerve, because it is just such a beautiful model. So we might actually, we might have gone from 20 years ago being behind everybody else to 20 years time we might be ahead of everybody else, just due to the beauty of how we can use the eye as, as the proverbial window to the brain, even though I hate using that term. Okay, second one, and maybe I need to hide behind my chair for this. I try and say this. I was about to say I should say this in the most polite way possible. Maybe I don't need to be too polite. The Australian system is a lottery that is frankly very broken beyond belief. The reason I say this, I'll give you the short answer, then I'll try and maybe explain it a bit better, because I think it's worth explaining, is that we can all criticize that funding rates are low, right? Okay, and that has to be dealt with at a governmental level. Oh dear. Hello? At a governmental level.
That's the government calling.
Yeah, that's the government calling.
That's the government calling.
That has to be dealt with at a governmental level, which is something we don't have kind of immediate power over, even though we can try and veto for that. Something that we can actively change, which I think is actually the structural problem for ophthalmology, is that Australia got rid of sections and panels for review. So those of you in the audience who are not academics, when you submit a grant, if you're in the UK or the US or just about any other country, if you write a grant on cancer, it goes to a cancer section who look at it, and then they send it to cancer researchers to review it, and then it goes to a panel of cancer reviewers that say whether it's a good grant or not, and then they can tell you if you got money. And Australia decided 10 years ago or so that they were going to get rid of this. So now if I write a grant in ophthalmology, it does not go to an ophthalmologist, it goes to a random person. Okay, fine. That means you've got to really sell your science really well. If I take any of you and you talk to your family, let's do this. Please just humour me. Put your hand up if you've heard of diabetes. Come on. I want to see every single hand go up. In you, Isabel. I'm looking at you. Put your hand up if you've heard of cancer, right? All right. Put your hand up if you've heard of mitochondrial optic neuropathies. Right, okay. You see where I'm going with this. So you can get more and more niche. If I write a niche grant in my area and it goes out to someone who doesn't understand what I'm doing, I'm never going to get a good score. And unfortunately, a lot of ophthalmology research is not sexy. And so I think we really need a structural change that allows, it's very unfair to people in certain sectors of research, because it means we're chronically underfunded because we're just not in the public eye enough. That's really what makes, so sorry, you said the first part of your question was because I've done this in multiple different countries. Okay. So to put this in perspective, last year it cost me a little over 2 million Australian to run my lab. 9% of that came from government grants, right? And I have a very high success rate with government grants. So that shows you just how difficult it is to run a successful operation off the government alone. If you want to really drive innovation, it's almost impossible. And like Flora said, the initial B3 clinical trials that we did were completely funded outside of government funding packages. So. And on that depressing bombshell.
and it sounds a bit dire, but at the same time, I really thank some of the advocacy that's happening in our sector. And I know there are some representatives that are in our audience, including Michelle from Amory, and things that we're doing here at CERA as well, to try and really advocate for medical research in general. And I think that it is difficult, but at the same time, we're really trying, and we're trying to do a lot of work in this area to try and push for that. And that's how philanthropy plays a big role in our work still, and for medical researchers, institutes around Australia, to make this kind of research possible and be able to translate these findings from the clinic and into clinical trials and into things that we can actually deliver and give to patients to help improve their eyesight and improve their lives. And so I think do we have time for one more, Georgia? She's got one short one. I've got one short one.
I have a question for Pete. Thank you for the excellent talk. I remember at the start of the lecture you mentioned that glaucoma is also heterogeneous, so there's a genetic factor that plays into glaucoma as well. So do you believe that these metabolic therapies influences accessibility to different people, or like the wider glaucoma patients with different
Okay, great question. I'll try and answer it quickly in two ways. And firstly, from all the different studies that our group, other people like Ian at CERA have done in the past, it's probably around 25 to 30% of glaucoma patients have a systemic or innate metabolic dysfunction that we've identified. That doesn't mean only that 30% will benefit from it, but it means that there's definitely that 30% that are down there. If we come back to our large genomic studies, you can do something called PRS, polygenic risk score. So this is taking basically the weight of the 129 mutations that you might possibly have as a glaucoma patient and going, if you've got these ones, you're over here in this group where you're very unlikely to have a severe glaucoma, and you might be in this group down here where it's a very severe glaucoma. And right now, they're usually ranking it around 8 groups, something like that, 8 very severe, 1 not very severe. You can actually then subdivide those into just the genes that are for the metabolic components of cells, but there's about 25 or 30 of those genes are specifically metabolic genes, and then make a metabolic weighted risk score as well. So we actually have the opportunity that if these kind of treatments work, we could do genetic testing and try and identify the right patients. What I didn't say, but it's important for the big TGNT, the glaucoma nicotinamide clinical trial, we're actually doing blood work of all the patients as well. So that the end here is that we will identify blood signatures of patients that progress slowly or quickly, or patients that are responsive or non-responsive to the treatment. So hopefully what that means is if this does work and becomes mainstream, we should be able to do a blood test where we should be able to look at the metabolic profile and say, you are a good fit, yes or no, and look at the genetic profile, are you a good fit, yes or no?
Thank you.
Thank you, Pete. And with that, I'd like to wrap up. So, thank you so much for being part of this, and I hope you enjoyed the panel. And I would also love to say thank you, Lani, for so much for sharing your story. And I love the fact that you've got your Ray-Ban Meta glasses on, because, again, it's a show of how a lot of research went into developing that, and now she's wearing it as something that can actually help her in her day-to-day life. So it just shows how you can translate something into a product that people can have and actually improve their life. So on that note, thank you so much, and I will pass it back to Keith.
Well, thanks once again to our panelists, to Flora, to Lani, to Pete. That was a great discussion. It could go on, I think, and we could talk about many of these issues long into the night. I think it's been a great evening, and as I mentioned earlier, the Crock family are an integral part of the CERA community, and this lecture every year celebrates the achievements of Gerald. But I'd like to now invite Tricia Crock, who, like Pete, has conducted research at the Karolinska Institutet. Many of you may not know that. Not at all. But and to present a commemorative plaque to Pete. And if you still haven't had enough of Pete, I understand he was interviewed by Vision Australia Radio this afternoon and is being broadcast at 8 p.m. this evening. So you can go home and listen to that as well if you still haven't had enough. But Tricia, would you like to come up and join us? And Pete, we'd like you to receive a plaque, and we'll take some photos.
Say a few words?
Yes, please.
Yes. Well, I think Dad would be absolutely thrilled, and I'm sure he's looking down and saying what a fantastic lecture this was. But I just wanted to make a couple of personal comments. The first is that you went from Cardiff to Bar Harbour, which is in Maine on the coast, and Henry Crock, who is our forebear, left St. John's, New Brunswick. He actually came from Baie des Chaleurs in Gaspésie in Quebec, but he went to St. John's, New Brunswick, which is literally up the road, isn't it, on the coast?
Like 60 kilometres away or something.
Yeah, so Henry Crock got the boat in 1850s to come out and find gold in Australia. And then the other side of our family, the Bladen side, my mother was a Bladen, they came from Stockholm. And yeah, so there's a sort of Swedish connection. And then
it's a Twilight Zone, isn't it?
Yes. So then I went back and I did some of my pediatric endocrine training at the Karolinska, and then subsequently did my pituitary autoimmune work at the Karolinska, which I'm still doing with them. So yeah, it's a few connections, but a Dad would be absolutely thrilled. And I'd also like to say is that still working? is I'm a co-trustee of the Dorothy Edols Trust, and Dorothy Edols was a patient with glaucoma, and I think it's absolutely fantastic that you have this consumer involvement. I think that's absolutely brilliant. And she gave Dad $1 million many years ago. It's now multi-million-dollar trust, which we give the money from the interest rates. So we want interest rates to be higher because then we can give you more money for the lab. And Dorothy Edols, I went to visit her at home, and she was very passionate about trying to do that. So she, up in heaven as well, would be so thrilled with just the changes that you're bringing to the field and the advancements. And I love it that you have so many collaborators, because that's the way in life to do things. It's not... And I know a lot of scientists have always try and beat other scientists, and they want to be first on the paper, and it's all very cutthroat, but this is the exact opposite, and that's why you're making such huge advances, and you're involving the patients as well. So I just think it's the best thing, and Dad would just be so happy. So we have a special thing to award you. I don't know what you call it. It's not really a plaque, but yeah, it is a plaque, isn't it? But it's in... So, yes.
Okay, well, that's it, everyone. So thank you so much for joining us. Again, we appreciate all your support and interest in the work that we do. Thank you for coming out tonight, and thanks to everyone who's watching online. You can catch up with the lecture online, so if anybody else missed it, they can see it. And please enjoy the rest of your evening. Thank you.